So thank you everyone for who has been sticking around with us until the last day of the workshop. So now just a reminder, as always, all the resources are going to be available on our website. So this is just a screenshot of the resource page of our website. And on the section CNSP 2022 tutorials, you are going to see, to keep seeing for the coming month anyway, the links to the skeleton, to the library. So these first two links are going to be updated. So, you know, and we will notify you whenever we are updating this, but we'll try to keep this compatible anyway. So, anyway, these first two links will be changed over time, will be updated. Then the beginner tutorials and the intermediate tutorials are here. We will put another set of links for the videos, for the recordings. So for example, I was on the second day, so yesterday I was with Mike, and I really want to watch also Joshua's tutorial because I've heard it was amazing. And I'm really looking forward to watch that. So we are going to put the link somewhere here. And you can follow that. So also their tutorials, so Joshua's and Mike's are going to be available in the future. So you'll still have the link here. You don't need to download anything now. So we're at day 3. This is a particular day, not a usual situation. Because, you know, the first 2 days were very practical and very on the particular methods that we usually care about for our studies, and a little bit of new findings from our keynote speakers, especially. And now we are at day 3. So the program for today is that we're going to have an invited talk first, then we have a little 5-minute break. Now, we are in the webinar setting at the moment. We will transition for the discussion to a traditional Zoom meeting. So there is a second link that I sent all of you earlier on today or yesterday for some of you. And so the discussion is going to be grouped in 2 parts. The first part that is a follow-up of Giacomo's talk on open science. And the second part that is more on current challenges with TRF analysis. And, you know, we'll talk more about that, of course, later. But, you know, issues when you have little data, or when the data is very noisy, or different or portable systems, or anything that has to do with current challenges and TRF analysis. And finally, Q&A, which is about whatever you want to talk about. So if you have questions on your data set, on some studies you're doing, and you would like to integrate our resources with your studies, or you have something that doesn't work, and you know, you can ask us anything. And we'll also take some time to clarify a little bit the call for projects. So if some of you have intends to apply for it, that's a good time to ask your questions. So today we are super happy to have Giacomo Baruzzo with us. He is a postdoctoral research fellow at the University of Padova, and his talk is about data standardization and sharing. And the title that we put is a lesson from bioinformatics, because Giacomo is in fact from a different field, a different area of research. His area of research is farther ahead in terms of open science than our field, way ahead of us. And so this is an opportunity for us, I think, to understand what the good things and the bad things are in his field in terms of open science, and so we can try to take the good side of the approach. So Giacomo works at the Department of Information Engineering at the University of Padova. His research is aimed at developing algorithms, methods, and software for the processing and analysis of biological data, from high-throughput sequencing technologies, especially single-cell RNA sequencing, bulk RNA sequencing, DNA sequencing, and 16S rDNA sequencing, which all sound complicated and exciting. Every time I see DNA, I think, oh, that's super cool. I suppose the same way that when you present your work and you say, oh, it's brain stuff, people are like, oh, brain stuff, that's cool. And so it's a different type of cool, and I'm looking forward to hearing your presentation, Giacomo. So I'll stop sharing my screen, and the floor is yours.
Thank you, Giovanni, and thanks to all the organizers for giving the opportunity to be here today. So I'm going to share my screen and my presentation. Now you should be able to see the first slide, right?
Yep.
Great. So good afternoon again to everyone. And in this presentation, as Giovanni already said, I'm going to tell you something about how it works, data standardization and data sharing in bioinformatics. So first of all, who I am and why I'm here. So I come from a different research field, as Giovanni said. So I work in the field that is called bioinformatics, and in particular in a research group from the University of Padua that is called System Biology and Bioinformatics Group. About my background, I'm an engineer. So I have a master's degree in computer engineering, and in my PhD, I moved to the bioengineering field, more specifically in the field of bioinformatics. So why I think that bioinformatics could have some common element to many of the people that are here today? Well, because one of the definitions that you can provide to define the terms bioinformatics is just a research field. It is very interdisciplinary, meaning that to do bioinformatics, you need some knowledge from computer science, biotechnology, engineering, mathematics, data analysis, etc. And is a field aimed at developing and assessing algorithm, data structure, method, and software with two main goals: the preprocessing first, and then the processing, or if you want, the analysis of, in our field, what is called omics data. So genomics, DNA, transcriptomics, RNA, proteomics, protein, and so on. Data that are generated by different technology, and each technology gives to the data different characteristics, different error, noise, and so on. So I guess that for the many of the people that are here in this Zoom link, if you just change omics data with your kind of data and just put the particular technology that are adopted in your field, well, this kind of definition that is more, if you want, data science, data analysis oriented, could be fit quite well, even some of your tasks. So for this reason, I guess that the things that I'm going to talk about today, which is basically tell you how it is the life, the everyday life of a bioinformatician, it could be useful even for other research fields. So I'm going to present some of the best practice or just some mandatory policies of what we did in our research activity with a focus on data and software. In particular, in this presentation, 80% is about data and 20% is about software. And don't be scared, because hopefully no background is needed in terms of molecular biology and biotechnology. So I will not tell the word DNA, cell, or molecule anymore in this presentation, at least hopefully. So what about the first part, which is also the part that gives the name to this talk? So the data. So how Giovanni said, there is some differences between research field in terms of data sharing, open access, how data are released, the format of the data, etc. But I think that the most important point is about data sharing, because it's really the key element that is important in any research field. So in this sense, we are not talking anymore about bioinformatics. We are just talking about releasing in an open format the data of our research. And I guess that we all agree, or many of us at least agree, that sharing data is good. But to be realistic, we also know that there are some elements that we should consider when we talk about sharing data. That typically people find these three terms important: money, effort, and privacy. So we all know that collecting and producing data is expensive. And so why should I release my data if they cost me a lot? This cost could be both in terms of money, but very often is it in terms of effort. It may be difficult to find the sample, difficult to enroll maybe the patient if data come from clinical trial or similar stuff. And there is effort in then collecting the data, processing them, analyzing. And depending on the field, there could even be some issue related to privacy or anonymization of the data, etc. So for this reason, I put the stuff here as the elephant in the room, because these three elements are often used as a reason to don't share the data. But I hope that I will show you that it's possible to overcome this limitation, to mitigate these three elements here. And as an example, I will show you what we did in bioinformatics in terms of data sharing. To do this, I will start first of all with the most famous example of open data in bioinformatics, so the case study of the Human Genome Project. Then I move to what is required to publish something in the bioinformatics field. So just a brief overview of what are the policy, the requirements that for us is mandatory to follow to be able to publish something. Then we are going to move to the two things that really makes data sharing possible: infrastructure and standardization or data format. These are two technical aspects, but are really the building blocks to make data shareable. Then I'm going to show you some example of extra effort done by the bioinformatics community in terms of data sharing. And I will conclude with this first part about data with some hint about how you can publish your data even without having analyzed them yet. But starting from the first part. So I think that we all know somehow the huge breakthrough that there was around 2000 when the human being was able to first publish the sequence of the human DNA, the human gene. The Human Genome Project is a project that started in the 90s and just end in 2004 with the publication of a very famous article, and together with the sequence of the human DNA, roughly speaking, a very huge string of A, C, G, and T that represent our genome. So the good things, I think, about the Human Genome Project is that since the beginning, open data and open science was one of the criteria in mind about the Human Genome Project consortium. And I think that a significant step it happened in 1999 when the first draft, so the first sequence, but with a lot of error, was available. And together with a paper when they provided first somehow characteristics, even in a very rough way, that we were able to identify about the human genome, they make this statement here that I put in the slide. So the member of the consortium used this chance, the publication, and all the media that were focused on this event to reaffirm that they want that the data are free and immediately available without any restriction. So this is, I think, a key point, because in 2004, when the paper comes out, few days after, probably a few weeks, the sequence was available. The sequence, the metadata, and all the protocol to generate that huge results for the human being. So this is an example that I used to say that probably bioinformatics is a field that was born in somehow in an open science framework, in an open data framework, because data sharing was something that was important from the founding fathers, if you want, of our field. So we were born somehow in an open science and an open data field. And this is even quite evident if you go to the biggest journal, the one that are more famous, higher impact factor, so choose the definition that you prefer, and you go to the data availability section in terms of bioinformatics data, so omics data, DNA, RNA, and so on. So for us, it's not a choice, it's somehow mandatory to release the data. And this is the first key point, is the policy and the requirement overjournal. That I think is a very important element to make a field, research field, open science and open data. So this is just a screenshot. I hope that you see well in your monitor of nature. So if you go to the link that we put on the bottom left part of the slide, you can see this screenshot here, this text here. And is the instruction for how to submit and where to submit the data. So as you can see, nature just divides the possible data that should be published together with the article in 10 main types of data. And the funny thing is that 7 over 10 are data related to bioinformatics, are omics data. And you see that there is the type of data that is mandatory deposition, so not an option, and the suggested or suitable repositories of data infrastructure when the researcher should put their data. So as you can imagine, it's not a choice. You should just choose. You can just choose your favorite repository, but it's mandatory that if your data fits one of the 10 descriptions here, then you are forced to share your data. This is Nature, another big journal, as they say today, Science. So here they classify the data in 7 main types of data. And again, 2 of them are data from the omics field, from the bioinformatics field. And still there is the suggested repository where we should submit the data. Another example, the last one, Cell, another very important journal. Here they just find 3 main types of data. And again, funny things, 3 are data from the omics field, from the bioinformatics field. So here, Cell, it provides additional layer if you want information. Because as you can see here from the screenshot, they don't only tell you that the data should be published, but they even tell you that there is some standardized data type. And the text here that you can see, this acronym here, BAM, SFF, and so on, are file format. So the journal is suggesting even the format of the data. And then in another part of the rules of a journal, there is obviously the list of the data repository where you should submit the data. So the policy of the journal somehow force the researcher to have some guidelines in terms of the format of the data, so the extension of the file, the format on which information are saved, and where to store the data. So in data infrastructure, in repository. And there is even some lists that are suggested or mandatory, but there is quite a clear indication on where data should be put. And again, really, file format and the availability of repository is really the 2 key elements to have open data. So the bioinformatics field, I put this slide here, probably you're not able to read the name of this repository, but just to give you an idea of the quantity of repository, each one for a different type of data that are available in the bioinformatics field. So obviously I have no time and probably it's not interesting to look at all of them. I just want to make an example of one probably of the most famous one. This infrastructure called SRA, which is the de facto standard repository for sequencing data. Roughly speaking, data that just string with A, C, G, T that you obtain from your experiment. This is a data infrastructure when you should upload both data and metadata. So data, again, the string of characters that represent the DNA, RNA, whatever, but even, and very important, metadata. So information about the sample, how it was collected, from which organism, and so on. This kind of platform are both very useful, and the typical application is just to retrieve data. So collect data produced by other research groups from other experiments. But also users are free. You just need to log in, and so to create an account and log into a website to upload the data from your experiment. Obviously, following some rules in terms of format and which kind of information you should provide. So this kind of data infrastructure, again, this is the example of the SRA repository, but there are some common concepts that are key concept, I think, when we talk about data sharing. And it's the fact that data sharing, data repository, in the bioinformatics field are made by 2 main elements. The repository itself, which is typically a website, when the user through a graphic interface just open a browser, can download browse, query all the available data. But also, and very important, some software tools. Typically, this kind of repository have the official suite of tools, some libraries in some programming language that allow you to programmatically query, download, convert, and do some processing on the data that are stored in this data repository. So it's the union of these two elements that makes the data sharing infrastructure. A website, typically, and a software, a library that is released to programmatically interact with the repository. So these repositories are a lot used. On the plot on the right side, you see the amount in terms of petabase, so base is just the single character in the string of DNA that are stored in this kind of platform, in particular in the repository called SRA, so the topic of this slide. And you see here from the number that there is a huge amount of data. When you see here runs is somehow a synonymous of experiment. And from the amount of data, you see that the community is always increasing its open data philosophy, because the amount of data that is produced is always increasing in the last year. So this kind of tools are also very useful, because when some emergency rise, for example, the COVID pandemic, well, there's such huge amount of data. Since they are there, they are organized in a very precise way with metadata, with a very precise file format. Well, it's quite easy to query, to realize this kind of data. And for example, the NCBI, that is the organization that is taking care of managing and handling this kind of repository, developed this tool that automatically, each time you upload a sample, if you want, you can run in background a tool that checks if there is some kind of COVID-related signal in your sample. So this kind of repository at the beginning was founded by NCBI, and so something related to the United States government. Since the amount of data is increasing a lot, as NIH and NCBI require always more and more money to handle this kind of databases. And so, for example, a contribution to this repository, to handle this repository, to buy, roughly speaking, the server and handle all the computational power that are needed to query to 25.6 petabytes of data, for example, in the last year comes from Amazon. So Amazon, for free, within the Open Data Sponsorship Program, support this kind of public repository that, again, in the last year becomes always and always more difficult to handle just from an institution that is funded just by one country. And so in this case, a private company, but through a Open Data Sponsorship Program, help to support this kind of research. So what it means for a researcher having this kind of data repository? Well, if you just open the SRA is a website freely available on the web, you obtain a page that looks like this. You have here a field when you can put your query. I just put cancer and just press enter. And this is what the website shows you. So you see there is a lot of data. Here is the list of all the samples stored in this database. It is, as you can see here, more than 500,000 that are somehow related to cancer. But very important, there is not only data, as I told you before, but there is even metadata. And indeed, these are all the information that you see here in this web page. So, for example, you see that more than 500,000 are open access data, while for others you need to sign some document, etc. There is some biological info. So this is telling that this cancer-related sample comes from human, mice, etc. There is the technical information, which instrument, which protocol were used to collect this sample. And you see here, if you are familiar with sequencing, the major vendor of a sequencer machine and protocols. There is information about the file format, so the data they're going to download, which file format we follow. And very important, you can do more advanced query than just type cancer and press enter, because you can use logical operator to do more advanced query on this kind of data. So this is just the first half of the infrastructure, the website. And as you can imagine, all this operation and even more, you can do with the software suite. So with the library that comes together with this service that allow the user to query this dataset and do even more advanced research and search, sorry, in this data. So this is an example of, again, one of the most famous data repository that makes the life a lot easier for research in our field. You just type cancer and you have more than 500,000 of sample associated with cancer that you can reanalyze, use to develop your new tool, or find new insight. Again, this is just, at least in my opinion, half of the things that are needed to do data sharing, the infrastructure. But the other half is having data stored in a uniform and clear way. So having some standardization in the data and in the format. So in the bioinformatics field, you can find two main kinds of standard of format. What I call hard data format. So the kind of format that you basically need to follow and to use in your tool, in your method, or in your analysis. Because if you don't use this kind of format, it's very likely you are not even able to publish your research. So an example here is a format that is called SAM or PEM. You don't need to catch the technical detail, but I think it is important the history, so why this format was proposed. So around 2008, 2009, there was a new generation of sequencing technology, so a new generation of measurement tool that allow you to measure DNA and RNA, roughly speaking. So this produced new kind of data. And to handle this kind of data, obviously, the research community started to develop a variety of tools. The problem, well, each tool produces the output in a different format. So even if they are solving the same problem, the output of this tool are different. So this is really a nightmare for the people that should take this data and do the next step of the analysis, because there is a variety of formats. And so each time you use a different tool, you need to be compliant with that standard data format and then change your pipeline. So for this reason, a set of experts tried to define a file format. And this was done in 2009 in this article here. But again, as always in the bioinformatics field, data come with software. So we release both the file format specification and a suite, a library, a piece of software that allow you to query, to convert, to compress, and to do many operations in this kind of file. Results, after basically a couple of years, 99% would say all the tools that did this kind of processing on the data were compliant with this format. And this enabled many, many good things. For example, a lot of benchmarking study rise in the field, because once you have a very homogeneous way to handle data, now you can focus first on developing your method, because you put your effort in doing a good analysis, not in just converting and handling data in different formats. Once you have method, you can benchmark them. And again, having a uniform file format is a lot easier to do this kind of stuff. But you can even do some challenges. So just to put a data where you have some sort of ground truth, provide the data in this format, and know that all the people that want to develop a new method, well, they should not be worried about the format, which is standard. We should just focus to find the answer on your particular question. And so if you think that this kind of article is quite boring or useless, well, just check the amount of citation that this article had. So this is really a breakthrough in the field. This kind of data and this kind of processing, the results that is then sent in this file format, is one of the first steps in the analysis of the great majority of omics data. And so this article have a huge impact in simplifying the life of researching the bioinformatics field. Again, this kind of format are hard formats, so all the tools follow nowadays this format. But there are even some kind of data, standardization data format, that we could define as soft data format. So something that is very used, is supported by many analysis pipeline, but still is not somehow mandatory, just suggested. So an example of this second class of file format, of soft data format, that I put here in this slide, is a R package that called single cell experiment. Again, all this presentation is about examples, so these are just one of the many examples that I could have done, but time is limited, and I put something that I think is relevant. So it's just an R package that contains two main things: a data format in the form of a class, so a class in the object-oriented programming way, and a set of method of procedure that allow you to store and retrieve data inside this object, as well as a set of methods that interact with some field that contain the primary data, some metadata, some, for example, the dimension of a dimensionality reduction technique that you can apply on this data. So even some metadata, if you want. And again, this kind of standardized way to handle data makes a lot easier the life of research. So this is a quite recent article published in Nature Methods just a year and a half ago. And you can see that it had already a quite good impact in the field. So this is just to tell you that infrastructure data repository comes with format of a data. And format of a data are necessary to have data in that infrastructure organized in a homogeneous way where you can both store data and metadata. So having this powerful ally that is open data in the field, where the community could build a lot of new things over this data. You can just, for example, reanalyze data that are there. You can just, if you are a method developer, you have plenty of data where you can test your hypothesis. Or if you are more oriented to the biological results, well, you have a lot of data set about maybe the disease or a particular biological process that you study that you can just reanalyze. But research community do even more with this kind of data. So for example, there is a lot of projects that are aimed to just take a subset of this data that could be related to a particular disease or to a particular biological process and do something more on this data. For example, the Recount project that I put here as an example in this slide is a data repository where the researchers that work on this project just collect samples from different studies. All the samples are obviously freely available in this data repository and apply a common preprocessing to this data. So the idea is that in these 3 iterations of a project, the first release was 2011, then 2019 and 2021. This researcher collect, as you can see, an increasing amount of sample from public database, starting from 400 up to almost 1 million, coming from different studies. They study from 18 studies, and now they have something like 18K studies. And they apply a common preprocessing. The picture here on the right is just a workflow, all the steps that they apply to say, okay, we preprocess the data in a uniform way. But the result is that if you now connect to this website, you have this amount of sample coming from this amount of studies that are all processed in a uniform way. So see, this is the best things for people like me that are method developers. So I'm not the guy that maybe find a new interesting genes. I'm the one that developed the software that a biologist then can use to find a new interesting genes. And so this kind of effort of the community is really, really very useful. And again, as always, data come with software, because the Recount project is both a website where you can again interactively browse the data, navigate on the website and do some query, but it's also a package, a piece of software where you can programmatically query the data, do some conversion, and have a more powerful way to interact with this huge amount of uniformly processed data. So this is an example of what the community build over the fact that this field is open data, is open science. And another very good thing, at least in my opinion, that the research community did is organize challenges on this data. So this is one of organization, the DREAM organization. This is one of the most famous one in the field of organizing open data challenge. So typically this is the workflow. You collect some data from a particular disease or from a particular biological process of interest, or even you are a group that you are developing, preparing, measuring new data. Well, you can collect this data and propose a challenge on this data that is typically try to solve a biological question. And then this kind of organization take care of providing the website, the rules, etc., to make different research groups try to answer to this question on this new data. And if the data are new, the second after the paper is published, the data are released to all the community, not only to the participants of this kind of challenge. in the last year, something that is not typical of bioinformatics, but is more broad, is the fact that even if you are not analyzing the data, but you are just producing data, but you have a way to just publish your data without the need to do any analysis. An example that is probably quite famous, not only in the bioinformatics field, is the journal called Scientific Data, where if your data follow some of these criteria, or to be more precise, all these 6 principles, then you're able to publish just your data. And typically the metadata, the information how you collect, but you don't need to do even the analysis on this kind of data. So this conclude quite quick, even if hopefully clear overview of what it means working with data in the bioinformatics field. And would like to spend just the last 5 minutes about software, even less than 5 minutes, which is the other side, if you want, of open science in bioinformatics. And I think that the important paradigm shift that we are still doing in bioinformatics is moving from computing to scientific computing. So the idea that you have to design your software using the right data structure, algorithm, heuristic, etc., even right programming language. For example, in bioinformatics, we tend to don't use programming languages that have license. Roughly speaking, 99% of the tool are not MATLAB in our field. You should always take care of the user. So the researchers that are going to use your tool, or the biologist that is going to use the results of your tool to find new biological findings, well, the code should be easy to run, well documented, having some test data, tutorial, etc. And important, very important in the last year, is to ensure the reproducibility, reusability, and accessibility of the results. And this is done having good software and always releasing the data. It's also related to be able, especially in the bioinformatics field, this is mandatory since we are handling what is called nowadays big data, to be able to exploit even modern hardware architecture and infrastructure. And to do just an example of what it means, at least in my opinion, doing good scientific computing, I just put some screenshots of the rules to be able to publish in this journal that is called Bioinformatics. So it's just, again, some screenshot from the rules that authors need to follow to be able to publish. And so you see that if you publish some piece of software, the author must provide a self-contained and easy-to-use software together with test data and documentation how to acquire, install, and run the software. So the importance of documenting your piece of code. The software should be freely available on stable URL, meaning that one time we tried to put our software in my personal web page, we got a major revision that they say, okay, the software is good, but if you don't put your software on GitHub, on GitLab, or whatever, we don't allow you to publish your software. And even in terms of software, is something that is always changing, but the version that you submit together with the article should be put in another URL. So not in GitLab, but for example, in Zenodo or in other repository where you can have a DOI and a more precise tracking of all the changes that happen in your software. This is for a piece of software, but here are the rules, even if you provide a web app. So if you are developing a web service or a website. And again, you should provide help pages, tutorial, support website for some years, and so on. So these are mandatory stuff. All the things highlighted in red are mandatory. And then there is some suggestions. So the authors are encouraged to release the software in open source license. In my experience, 99% of the paper in bioinformatics are open source. So this is more than encouraging, is mandatory. And to don't use proprietary programming language. For example, this is the reason because MATLAB is not very common in bioinformatics. And provide the code to reproduce the results always that you put in your manuscript. So this is just, again, the rule that we need to follow to be able to publish a software. And if you take the things that are highlighted here and come back to the previous slide, well, you realize that many of the things that good scientific computing are good scientific computing are really the requirements that we have to follow to be able to publish in bioinformatics. So in terms of reproducibility, reusability, and accessibility, this is just a pointer. In the last year in bioinformatics, we find software categorization very useful. So I don't want to go into detail here, but if you are struggling with the problem of ensuring your software very high reproducibility, reusability, and accessibility, the keyword that I suggest you to Google is software containerization. So last slide is, while open science, in my opinion, is no more something that is a good thing, but is somehow mandatory. And I think that is something that the European Union, with the last funding framework, which is called Horizon Europe, tried to somehow force or to put as a hard requirement. Indeed, if you're just looking at the document to try to get funding from the European Union, you will see that open science is a key element across all the programs, meaning that independently if you're going to apply for life science or for an industry project, open science is something that should be there somehow in your project. So just to recap, what I hope that I show you in this presentation, how data and software sharing works in bioinformatics. We saw the important role of having some, if you want, hard or quite strict policy and requirements in terms of journal, because force research to follow best practice, meaning following some specific data format, put the data in the right repository, and produce good software. We saw that it's fundamental importance to have data and software sharing, having some infrastructure that allow you to not just to put the data in the software, but even to put metadata and to be able to do some query, both in the website and using some libraries or in a programmatic way. The fundamental role of having some standard. Data infrastructure comes with data standardization, and data standardization comes with data infrastructure. What the community could do, having this tool here, could build new data repository, pre-process the data in a uniform way, organize challenges and benchmark. And that is possible even to publish just your data, your code, thanks to new journal. In terms of coding, it's important to have a shift from computing to scientific computing. That it means, especially in the last year, taking care of the user, make the code easy to read, documented, provide test data, and make sure that your code together with your data make results reproducible, reusable, and accessible. So the take-home message, I hope that I show you that open science is the way to go, and is the way that in the bioinformatics field we are trying to use to make good science. And that sharing data and software is possible. I hope that I provide you some example where sharing data and sharing software is possible, and you should not think that our experiments are less expensive or that there is no effort or no privacy issue. But we find some tool that allow us to overcome somehow these three very important elements: the cost, the effort, and all the problems related to privacy and ethics. So I hope that I convince you that a possible open science framework, open science is a possible framework, and that in bioinformatics, more or less, we are trying to follow this kind of path. So this is the last slide of my presentation. I thank you for the attention, and I'm more than happy to answer to any question.
Thanks, Emilian. That was great. Very, very strong talk. And we have a private chat here between the organizers with a number of questions that are coming up. So for the participants, please feel free to put your question in the Q&A. We will have a proper discussion open to everyone to speak in a minute. But first of all, thanks, Emilian, Giacomo. This was very, very useful. And I see Nate there with the hands up. Let me just say something before I give you the microphone, Nate. One thing that struck me from your presentation, Giacomo, is that there are a couple of things. One is certainly our field of neurophysiology is different in many ways. And one thing that you said is that bioinformatics starts with an open science framework, while neurophysiology really has a long history. And so there is some resistance, and things are happening very fast now, more recently. Especially with the paradigm shift that this workshop is kind of trying to capture with the continuous type of sensory experiments. So there are some differences, all right. And so I suppose that our point is, let's try to capture the good things and the bad things. When I think about data sharing and I see from your presentation, there is a lot of work involved. And it's terrifying because I want to do the science. I don't want to spend time, you know, learning all of these things, and I can see the benefit. But when you show the path and your plots as well, which are somewhat similar to what I, you know, certain slides were similar to what I was discussing in the first day, you know, the inevitability of getting closer to that stage, not necessarily the same stage, it really struck me. So I think it's important that we stay ahead of our time rather than you know, staying behind. Nate, do you want to ask your question?
Sure. So actually, it is almost the same question as Bjorn's. So I'm going to bring his up. I generally wanted you to talk some more about the way bioinformatics deals with privacy. Bjorn also points out, as far as I'm aware, the General Data Protection Regulation of the EU states that data needs to be anonymized in order to be published, and there should be no way of re-identifying from whom the data was collected. So I was wondering if you can address that as well.
Yeah, so obviously I can, you know, talk about the bioinformatics and biological point of view. I'm not a lawyer, so I cannot provide all the details about the regulation, but it is that is the same obviously for us. And even before, you know, this kind of GDPR or any kind of rule, obviously, when you release the data, these servers, for example, are stored typically in the USA, so, you know, you should be compliant with the rule of the country that handle the server. Well, there is for sure a lot of attention in the privacy stuff. So something that I used to say, okay, guys, we are sharing DNA of people. You know, in the common idea, what is more a fingerprint than the DNA? But you know, you see there is 500,000 of sample there. So how we can do this stuff? Well, because, you know, really not all the, for example, genetic material that you can collect is a way to identify people. So I mean, there is a lot of you know, of variability in the kind of data and information that could be stored, to be present in a genomic sequence. So make it simple. If I just collect some genomic data from a guy, well, unless I cover some particular region, or I'm covering some other, let's say, biological information, this makes the sample not identifiable. So I'm not able to connect that sample to a particular person. In terms of, if you want, metadata and all the covariates that could be associated, well, this is quite similar, if you want, to the rules for clinical data. So even there, if you are collecting some biological information from, I don't know, exam that that the patient did or whatever, well, there could be a combination of features that could make a patient more or less identifiable. But, you know, the problem is that it should be 100% identifiable starting from the data. So I mean that the part of rule about the anonymization of patient, I guess that the law is the same from one from clinical data. The only difference here, it should be, you know, the amount of information that is encoded in the genomic data that could make that patient identifiable just starting from the genomic data. And again, depending on the kind of experiment, this information will be more or less present, but typically, you know, you haven't, especially with, I know I was thinking about RNA-seq or proteomics data, is, I would not say impossible, because I mean, I'm not both a biologist and a lawyer, but I mean, in my experience, it's quite easy that you have no issue about the identifiability of a person. Again, to be more precise, we should talk about a specific data, a specific genomic feature, etc. But I say that in general, I have the chance to collaborate with the production of at least 2 datasets, and the legal part was quite smooth, because for that kind of experiment, it was RNA-seq. It was basically impossible to associate the sample to a particular person.
And that could be a great point to continue in the discussion in a couple of minutes in terms of how that applies to our domain, because when you talk about EEG, MEG data, you know, there are some people that start saying how, oh, you know, it is identifiable data that should be considered personal data. And while honestly, I mean, it's much worse than DNA or RNA data in terms of identification, possible identification of the person. I'd like to continue the conversation in the discussion. There is another question in the chat there about who's funding those massive databases. I know that you mentioned a couple of things earlier on in the talk. Could you, like, if you had to run a new study, so that's also a question, like, you said that some labs, maybe they just collect data in that particular study and they share data. Maybe they don't do any science on it themselves. Who do you apply? Where do you get the funding for that?
So just to make an example, to best of my knowledge, this kind of stuff like the recount project is handled by just the computing facility of the research group that participate to this project. So it's not something financed by the NIH or something like that. Again, I should double check the paper, but you know, this size of data, even if it seems a huge amount of data, really is nothing compared to the SRA archive that I showed you before. In the case of this, worldwide repository like the SAA archive or the GEO archive, or there is even the European counterpart of this kind of archive, they are typically financed by, I mean, in US is NIH, plus in the last year some, big tech company that provide for free some support in terms of computing power, like Amazon. In terms of Europe, well, there is a lot of effort in the last year. I don't know if you are familiar with the Elixir Consortium, which is a organization, a consortium partially financed by the European Union that is in the field of life science. And one of the things that this consortium is providing to researchers is storage and computing power. So it's one of the assets that they provide. So if you, for example, need this kind of infrastructure, maybe you can try to apply to enter in one of these Elixir projects and find the right resources or even right funding for you. In the US, it's NIH and similar stuff. In Europe, we have the European Bioinformatics Institute, but again, is partially financed by some private institution, foundation, or mainly by some universities. So really, the resources of finding are quite different. But again, if we talk about this worldwide repository, so where all the community put the data, where we are probably, I don't know, 20 in the entire world, and by financed by part government, part research organization, and again, in the last year, even some private big tech company.
That's great. Thanks, Emilian. So we went a bit over time now. So what I would like to do is to have 5 minutes break, and then we log in the Zoom meeting, and there everyone has direct access to the microphone, and we can continue the conversation. The Q&A function is empty now. There are no more questions from the public, but we can talk more directly there, and we have a few discussion points for you, especially there, Giacomo, in the first part. So thanks a million again, Giacomo. That was brilliant presentation. So 5-minute break. You have the link to the Zoom meeting. I'll send it around again, and I'll see you in 5 minutes on the other room. All right. Thanks a million.